Hey, good morning. So we are in a week of October 26 through 29. So we're finishing up neural tissue. So next week we have a quiz over chapter 12, which we're covering this week. Okay. So what I will tell you is I'm still in the middle of grading exam 3. What I can say at this point is don't be so concerned about your grade initially on it, because as you probably could tell when you took it, there are a lot more short answers on this one than the last one. I will say, so far, that is helping you out a lot. So I haven't graded everybody, so I've graded a few, but for those that I have graded, there's been a huge swing between the grade before I graded the short answer and the grade after. So, don't look at that grade and assume, oh, I bombed it, because you very well may have done extremely well. Because also what I noticed is that in scanning how you all did on the multiple choice, you all actually did extremely well on the multiple choice. So it's just a matter of how many points on the short answer did you get. So just wanted to kind of put you at ease till we get those graded. Hopefully I'll be able to review with that on the next class, but we'll see how, since it's more short answer, it's more for me to grade. So just wanted to give you an update there. All right. Okay, so let's pick up here. I believe we finished just talking about the steps in the cholinergic response last time. Hopefully that kind of makes sense. Remember learning about acetylcholine and how that's involved in action potentials across a synapse. All right. So synaptic delay, delay just means there's a time period between when something happens and the next thing happens, right? So synaptic delay happens between the arrival of an action potential at the axon terminal and whatever effect it has on the postsynaptic membrane, okay? So think about it from that perspective. There's action potential at the axon terminal, then there's some effect in the postsynaptic membrane. And however long it takes for that to happen, that's your synaptic delay. And that takes between 0.2 and 0.5 milliseconds. Now, it's typically going to be due to how much time it takes for the calcium ions to basically move into the area and how long it takes for the neurotransmitter to be released. You remember, your calcium ions are your trigger for your axon terminals to release their neurotransmitter, right? So basically, it's one of the things that lead up to creating the neurotransmitter release. Now, another factor with the synaptic delay is, well, how many synapses are there? So if you had just one synapse, you're going to have a faster response than if you have multiple synapses, because each synapse means you're going to have to repeat that process. Okay. Now, we haven't talked about reflexes yet, but we will. But some reflexes will involve just one synapse. Others will have multiple. Okay. Some reflexes just have a afferent neuron and an efferent neuron. Some reflexes have an interneuron in the middle, which means there would be two synapses before you get to the effector cell. All right. Another concept is synaptic fatigue. Right? Fatigue just means you get tired, right? So let's think about it in the context of a synapse. So if neurotransmitter, if it's not being recycled fast enough to meet the demands of the stimuli that are around that axon, the response to that synapse is going to weaken, okay, until you get enough replenishment of acetylcholine. So y'all will recall in our steps here that we talked about with the cholinergic response, in that last step, acetylcholinesterase comes in and degrades acetylcholine to choline and acetate. Choline goes back or reabsorbs back into the axon terminal, and the mitochondrion provides acetyl-CoA, which is used to replenish acetylcholine. Okay, so if that process of recycling doesn't happen fast enough, you're going to get a weaker response in that synapse because you don't have enough acetylcholine. Okay. And so that synapse is said to be fatigued. So it's like if you want to run a race, but you don't have enough oxygen, you don't have enough nutrients in your body, you're going to get tired, right? Same for a synapse. If a synapse doesn't have its nutrient source, the acetylcholine, the synapse is going to be weaker. Okay. Let's take a break and discuss synaptic delay and synaptic fatigue with your classmates. All right. You can do this at home as well. All right, so let's talk a little bit about neurotransmitters and neuromodulators. Nervous system, right? So we talked about acetylcholine as a neurotransmitter, but let's talk about not so much specific ones, but classes. So neurotransmitters are said to be either excitatory or inhibitory. And, before you get caught up in big words with multiple syllables, just, think about it. So neurotransmitter, it's something that transmits some signal in your nervous system, right? Excitatory, this means it causes depolarization of the postsynaptic membrane. So remember, a neurotransmitter goes across the synaptic cleft, attaches to a receptor on the postsynaptic membrane. If it leads to depolarization, it's going to promote an action potential, and it's said to be excitatory. Now, if it's an inhibitory neurotransmitter, it causes hyperpolarization, okay? And at the postsynaptic membrane. And the effect of the hyperpolarization is to suppress the action potential, okay? Which should make sense because your membrane potential is going in the opposite direction, right? So, and I think I did it kind of like with my hands last time. You can also do it with your arms. So if y'all look up here real quick, you can do, essentially depolarization, repolarization, hyperpolarization, right? So that should make sense. The depolarization and hyperpolarization are going in opposite directions. So you're going to have an opposite effect, right? So okay, we're learning about these terms. Okay, well, what's the big deal? So what's the effect of that neurotransmitter on the postsynaptic membrane? Well, okay, here's your scientific answer. It depends, right? So it depends on the properties of the receptor, okay? So there's the neurotransmitter itself, then there's the receptor that it attaches to. And each of those have some say in what happens, okay? So it's like you have a key, the key's got to fit the lock, but the key and the lock together affect what happens on the other side of the door, okay? So it's not really, in a sense, it's really not the neurotransmitter itself, because the neurotransmitter typically isn't crossing the membrane. Okay, I want to make sure y'all are clear on that. So think of it like this. Like if I walk up to a wall or a gate on the neurotransmitter, and on the gate there is a board and it's got a bunch of buttons on it. Or let's just say it has 2 buttons on it. If I press one button, it's going to do one thing. If I press another button, it's going to do something else. I don't actually go on the other side. I just press the button. That's kind of what the neurotransmitter does. Okay. So it's basically kind of what happens. So it's really not so much the neurotransmitter itself, it's the neurotransmitter, basically connecting with the receptor. The receptor does what it does. All right. Because the receptor is sort of embedded in the membrane. Half of it's on the side where the neurotransmitter is, half of it's on the inside. Okay, and the half that's on the inside does something on the inside. So if we're going to describe neurotransmitters more by their chemical properties, first we're looking at function, like are they excitatory or inhibitory? Now we're looking at more of a chemical classification. So they can be known as biogenic amines. If you remember, we talked about amino acids having an amino group with a nitrogen, 2 hydrogens, NH2. Okay, so you can be an amine. You can be an actual whole amino acid. Okay, so just being an amine doesn't mean you have the other parts to make an amino acid. Okay, just not necessarily a carboxyl group. Okay, not necessarily a side chain. So amino acids would have all of that. Neuropeptide. Anybody remember what peptide means? What class of biomolecule is a peptide? Proteins, thank you. So it's a nervous system, or nerve peptide, nerve protein, and dissolved gases. Okay. So yes, gases can dissolve. All right, they can be mixed in with what's going on inside those nerves. Let's talk more about this first category, the biogenic amines. Okay, you may have heard of this one, norepinephrine. You may have also heard of dopamine. Okay, so we haven't really talked about adrenergic synapses, but they are released by adrenergic synapses. Okay, not the cholinergic. So their effect is excitatory. Also, going with that, they cause depolarization. You'll see them widely distributed across your brain, as well as portions of your autonomic nervous system. Okay, remember, autonomic has to do with your sympathetic and parasympathetic nervous systems, fight or flight, rest and digest. All right. Dopamine is strictly a central nervous system neurotransmitter, so just for brain and spinal cord. Whereas norepinephrine can be more widely distributed across your nervous system, even into the peripheral nervous system. Okay. Dopamine can be excitatory, but it can also be inhibitory, so that's a difference between dopamine and norepinephrine. Okay, dopamine actually is involved in Parkinson's disease. Okay, and it's also affected by use of drugs like cocaine. Okay. So, if you ever want to learn more about dopamine, there's a movie called Awakenings. It was back in the 1990s, so probably when you all were either A, not alive yet, or B, when you were younger. But talks a little bit about how dopamine and drugs that affect dopamine pathways affect people with neurological issues. So, an interesting movie. All right, neurotransmitters and neuromodulators again. So we're still on the biogenic amines. Another one is serotonin or serotonin. It's also a neurotransmitter of the central nervous system. So just the brain and spinal cord. Affects your emotional state, affects your ability to focus or have attention on things. Okay. So if you're alert and awake right now, serotonin pathway is working well. Okay. So another is GABA or gamma aminobutyric acid. Okay. You can see why we just call it GABA. It has an inhibitory effect. Okay. Now, in the central nervous system, we're still learning about what it actually does. We know some of what it does, but we're still trying to, researchers are still trying to figure that out. Okay. But, just know that those are in that category of biogenic amines. All right, neuromodulators. So first we've talked about neurotransmitters, and then we're going to talk about neuromodulators. But before we do that, let's take a break and talk about what neurotransmitters are and how they're classified. Okay, so let's do that. You can do this at home as well. Like I said, we've talked about neurotransmitters. Now let's talk about neuromodulators. So these are chemicals that get released at the axon terminals. Okay. But they are not neurotransmitters. So what do? Well, they can affect the rate at which the neurotransmitter is released. They can also affect the response by that postsynaptic cell. Okay, so they're not the neurotransmitter itself, but they can affect what happens with it. Effects are long-term, and they take a long time to appear. The responses have multiple steps involved, and they also have what we call intermediary compounds, which just basically means because there's multiple steps, there's lots of compounds or chemicals in the pathway. In the neuromodulators, they can affect what's happening at the presynaptic membrane, the postsynaptic membrane, or they can affect what happens in both membranes. And they can be released on their own, or they can be released with the neurotransmitter. So hopefully you can see why we covered neurotransmitters first, so you can kind of understand then how the neuromodulators affect what happens with it. So just like there were classes of neurotransmitters, there are classes of neuromodulators. All right, so here we find the neuropeptides, the nervous system proteins. So these are basically small peptide or protein chains. They're made and released at the axon terminal. Okay. Now this class of chemicals called opioids. And they bind to the same receptors as drugs like opium and morphine. Now, there's different classes of opioids. In your central nervous system, you have the enkephalins, the endorphins, and the dynorphins. Okay, and obviously we know in the US there's a big issue with opioid abuse and addiction. But if you can, the more you understand sort of how these neurotransmitter pathways work, you can start to understand why they're so addictive and why they're so destructive. Okay? All right, now we also have the dissolved gases, and they can also serve as important neurotransmitters. These are things like carbon monoxide and nitric oxide. Okay, so I know you may have heard about like carbon monoxide poisoning. Is there a certain amount of carbon monoxide that you may have in your body? Yes. Can it be too high? Yes. If it's too high, can that make you sick? Yes. Okay, so it's not that any carbon monoxide is bad. It's just that if you're, classic example of people breathing in too much exhaust. That can be very damaging, again, because it can not only affect how you breathe, but it can also affect your neurotransmitters. Okay, so just wanted you to be aware of that. So neurotransmitters as well as neuromodulators. Their effects can be either direct or indirect. So the effect can be direct on membrane potential, and that can happen through opening or closing these chemically gated ion channels. Okay, remember we talked about different kinds of channels last week, okay, in the previous videos. This is the chemically gated ion channels, okay, and that's what helps you get to depolarization, okay, because it's, the voltage gated ones are open once you reach your threshold potential, but the chemically gated ones typically are going to be open before that to get you to threshold. So example there, acetylcholine, glutamate, aspartate, those are different. Direct neurotransmitters. Now the ones that have an indirect effect through a class of proteins called G proteins. We include epinephrine and norepinephrine. So E is for epinephrine, N is for norepinephrine. Dopamine, serotonin, histamine, and GABA. Okay, and that, you can also have an indirect effect via intracellular enzymes. All right, and so direct means they're affecting what's happening themselves directly. The neurotransmitter and neuromodulator does it itself. If it's indirect, it affects something else, which affects what's happening at the synapse. So the indirect effect on the intracellular enzymes, those are your lipid-soluble gases or dissolved gases like carbon monoxide and nitric oxide. All right, so this shows you that in the picture. Here's your acetylcholine binding to your chemically gated ion channel, allowing your sodium ion chemically gated ion channel to open. So acetylcholine, glutamate, and aspartate are examples for the direct effects. Now for the indirect effects by the second messengers, you have your G proteins, and they can link your first messenger, which is your neurotransmitter, to your second messenger, which is the ions and molecules that are doing something inside the cell. Okay, so we call that basically the second messenger pathway, because the neurotransmitter is outside the neuron or outside the cell. Okay, the protein essentially the neurotransmitter attaches to the G protein, triggers something that happens to an ion or molecule inside the cell. So the G protein is the link between what happens outside the cell and what happens inside. G proteins have an enzyme in them, and it gets activated when that extracellular compound binds. Okay, an extracellular compound, the neurotransmitter, outside the cell. Example is adenylate cyclase, and it actually makes your second messenger that we call cyclic AMP. And remember, enzymes or classes of protein or type of protein catalyzes a chemical reaction. So it's changing something into something else. Okay. All right, so here's your indirect effects by the G proteins. Here's your neurotransmitter, attaches to the receptor, connected to a G protein. So here the G protein is inactive, but when the neurotransmitter binds to the receptor, it activates the G protein, which activates the enzyme adenylate cyclase, which converts ATP to cyclic AMP. A cyclic AMP can activate other enzymes that deal with metabolism. It can also act as a second messenger and open these other ion channels. Okay. So it's a secondary effect because the neurotransmitter isn't opening this channel. Does that make sense? It would be direct if the neurotransmitter opened this channel on its own. It goes through this whole process, so it indirectly opens the channel. All right, so let's compare that to this one. Here, the neurotransmitter binds, opens that specific channel. Whereas this one, it opens a different channel. So it's indirect. All right, so you can also have the indirect effects through those intracellular enzymes. All right, so you have the lipid-soluble gases, the carbon monoxide, nitric oxide, and they'll diffuse through those lipid membranes, phospholipid bilayer, the plasma membrane, and it will bind, those gases will bind to enzymes on the inside of cells in your brain. Okay. All right, so here's an image of what that looks like. Nitric oxide can actually diffuse across the plasma membrane, activate certain enzymes, and they can produce second messengers. Okay, those second messengers can open up other ion channels. Okay, again, this is indirect because the nitric oxide isn't interacting directly with the channel. Okay, goes through this secondary or indirect process. All right, so let's talk about the effects of neurotransmitters and neuromodulators. Okay, you can talk about that with your partners. Okay, so we talked a lot about the neurotransmitters and the neuromodulators. Now let's talk more generally about how information gets processed in your central nervous system. So when your postsynaptic cells responding to information from the presynaptic cell, it means there's going to be some integration of that stimuli. Okay, it's responding to it, integrating that information. So at the simplest level, that's going to happen through individual neurons. Now, remember we learned about parts of the neuron, the axon and the dendrites in the cell body, right? Remember that? Okay. So for a given neuron, you're going to have many dendrites for that neuron that are going to be receiving messages from the neurotransmitters simultaneously. Okay. Now, some of those messages are going to be excitatory responses. Some are going to be inhibitory. Okay, so what we're looking for here is what's the net effect, okay? So if you sum all of that together, what's the net effect of those messages? So the net effects are on the axon hillock. So remember, that's sort of like the base of the axon before it travels along to the axon terminal, is going to determine whether or not you get an action potential. Okay. So concept we want you to know are the postsynaptic potentials. So these are graded potentials, and they get developed in a postsynaptic cell. Remember, the graded potential is, are you getting enough of a potential to get toward threshold, right? But these are graded potentials in the postsynaptic cell, and that happens in response to some neurotransmitter, right? Now, there are generally two types of postsynaptic potentials. They can be excitatory or inhibitory. So EPSP or IPSP. So if you get a graded depolarization of the postsynaptic membrane, that's said to be an excitatory postsynaptic potential. Now, if you get a graded hyperpolarization of the postsynaptic membrane, it's said to be an inhibitory postsynaptic potential. All right, excitatory, inhibitory. All right, neuron is receiving many inhibitory postsynaptic potentials will be inhibited from producing an action potential. All right, because you're actually going to need more stimulation to get to a threshold at that point. So if you're essentially decreasing the membrane potential, let's say you start out negative 70 millivolts, remember that's resting membrane potential? So actually, if you inhibit that, or if you're causing hyperpolarization, it's going to go past that and be like negative 90 or 100. If you're trying to get to negative 60 or 55, now you have a further distance to go, right? So you'll need more stimulation. All right, now to trigger an action potential, typically it's not going to be enough just to have one excitatory postsynaptic potential. Now your EPSPs and IPSPs, or excitatory postsynaptic potentials and inhibitory postsynaptic potentials, combined through what we call summation, right? You're adding them together. And that summation could be temporal or spatial. Now, if you have temporal summation, it's typically going to involve one synapse, where you'll have rapid and repeated stimuli. Okay, so they're happening very fast and getting repeated over and over again. So that's temporal stimulation or temporal summation. For spatial summation, you have multiple synapses. And you're getting stimuli arriving simultaneously at more than one synapse. So for one, you have one synapse where you have lots of stimuli repeatedly hitting that synapse. One, you have multiple synapses where their stimuli are arriving at the same time, the multiple. So if this is my dendrites in my cell body, so if you had temporal, you basically have one synapse and you're just repeatedly, hitting that dendrite with stimulus. That's temporal summation. Whereas spatial summation would be simultaneously hitting multiple synapses same time. Okay. Now, it could be the same dendrite, but just different spots also. Okay, that would also be spatial summation. All right, so probably helpful to visualize that, right? So temporal summation happens on a membrane. You get two depolarizing stimuli from the same source in rapid succession. Okay, effects of that second stimulus are added to those of the first. So here's your initial segment, your axon hillock, getting your first stimulus there. All right, second stimulus arrives, and you're adding that to the first, and that allows the cell to reach threshold. Okay, now you get an action potential. Okay, so those essentially those two stimuli were added together. Now for spatial summation, this happens when you get two stimuli arriving at the same time, but at different locations. Okay, you have local currents or specific currents spreading these depolarizing effects. And there is an overlap, we're going to experience the combined effects. All right, so here you got two essentially axon terminals reaching this axon hillock. Okay, so they're getting two simultaneous stimuli. In this area of overlap, you get a strong membrane or a strong action potential. Well, you're getting a strong stimulus rather. And that strong stimulus, that's the combined stimulus, is enough to reach threshold, so you're going to propagate an action potential. Okay. So here you have, for temporal summation, repeated stimuli at the same location. Okay, now you have two stimuli at the same time. That's the difference. And if it helps to remember, there's a space between the two stimuli, so spatial summation. All right, so let's go ahead and discuss the difference between temporal and spatial summation. Okay, you can do that with partner. All right, so let's talk about more things related to information processing. So as the excitatory postsynaptic potentials accumulate, you get the membrane potential getting closer to threshold. Okay. We say that neuron is becoming facilitated. And it's facilitated until that small stimulus is able to trigger an action potential. So it's kind of like you're getting -70, you're getting -68 to 66 to 64, 62, 61. Then you really just need a small stimulus to get you to the -60. All right, so you can get to threshold. So let's talk a little more about summation of these excitatory postsynaptic potentials and inhibitory postsynaptic potentials. So we talked previously about neuromodulators. We also talked briefly about hormones, not a whole lot. We'll get more about hormones in AMP2. So they can change how sensitive the postsynaptic membrane is to neurotransmitters. And that can actually shift the balance between excitatory postsynaptic potentials and inhibitory postsynaptic potentials. So it's not just summing the excitatory and inhibitory potentials, you also have to take into account the hormones and the neuromodulators. All right, so here is figure showing interactions between excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Here's your resting membrane potential. Okay, then you're going to have a depolarizing stimulus applied. You get an excitatory postsynaptic potential. You remove the stimulus, you go back to resting membrane potential. Right, you get more of a repolarization. All right, back at resting membrane potential, this time you get a hyperpolarizing stimulus and you get an inhibitory postsynaptic potential. If you remove that stimulus, you go back to resting membrane potential. Okay, and at time 3, you're going to add both. Okay, and in this example, there's no change in the membrane potential, even after you remove the stimulus, because the summation, they just canceled each other out. Okay. All right, another concept here, you have what we call axo-axonic synapses. Okay, it sounds difficult to say, but if you break up axo and then axon, basically just means that there's a synapse between axons. Okay, for 2 different neurons. Okay, so that makes it axo-axonic. Now, if we're talking about the presynaptic membrane, you can have inhibition or facilitation. So if you're decreasing the rate of neurotransmitter release at the presynaptic membrane, that's said to be presynaptic inhibition. And if you're increasing the rate of neurotransmitter release at the presynaptic membrane, we call that presynaptic facilitation. So if you increase the rate, facilitation, decrease the rate, inhibition. Okay. All right. So here is an example of presynaptic inhibition. All right, so this is looking at this under higher magnification. So here's one axon terminal, here's another axon terminal. Okay, so this is axo-axonic, because an axon touching an axon, or synapsing with an axon. Now this axon is synapsing here at the postsynaptic cell. So this axon can affect what this axon does with this cell, or in this case, this axon, or this neuron. So if you have an action potential in the green axon terminal, you can release GABA, and GABA can inactivate calcium channels in this axon. Okay, so when you get an axon potential, while you would normally be opening these channels and releasing or having a lot of calcium ions go into this axon, GABA is binding to the receptors, and you get fewer calcium ions entering the cell. And if you have less calcium, it means you're not going to release as much neurotransmitter here. Because calcium could be triggered to release your neurotransmitter. And so with less neurotransmitter release, you have a reduced effect here at the postsynaptic membrane. Okay, so that's presynaptic inhibition. So if the yellow neuron, let's just say the yellow neuron is a salesperson and the purple neuron's a customer, if salesperson's a good salesperson, they're going to be able to, have an effect on customer. But, let's just say you're able to get the green one. Maybe there's an ad that says, hey, this one doesn't work as well as, this product doesn't work as well as the other. Therefore, this salesperson isn't going to be able to communicate its message as effectively. Okay, so it's essentially inhibiting this other neuron. Okay. All right, presynaptic facilitation. This is the opposite effect. So you have an action potential arriving, causing the release of serotonin out of the axon terminal. And serotonin actually is going to activate the calcium channel, so you get more calcium ions in it. And that's a trigger to release more neurotransmitter. Okay? Now, when this neuron gets its own action potential, so with more calcium ions, you get more neurotransmitter released, and you have an increased effect on the postsynaptic membrane. So this is the opposite. Now, information, if we're thinking about these nerve impulses as information, it can be conveyed just by the frequency of the action potentials that postsynaptic cells receiving. Now, what affects that? Well, the degree of depolarization above threshold. So if you're able to hold the membrane potential above threshold, that's almost like having the same effect as having another stimulus that's large. So being able to hold depolarization and stimulus helps your neurons process the information. Now, the maximum rate of your action potentials gets reached when you eliminate that relative refractory period. And if you remember back a few weeks ago, relative refractory period, no, last week rather, you have the absolute refractory period and the relative refractory period. In the absolute refractory period, nothing you do is going to be able to trigger another action potential. But in the relative refractory period, okay, because some of those channels are closed but capable of reopening, you get a larger than normal stimulus to trigger another action potential. Okay, so if you're able to eliminate, you have a strong enough stimulus, you're able to eliminate the relative refractory period. Okay, so let's kind of summarize what we're learning here. But before we do that, let's take a break and discuss with your partner differences between presynaptic inhibition and presynaptic facilitation. Okay, I'll give you a minute to do that. You can do this at home as well. All right, so let's summarize. So information in your nervous system gets relayed through action potentials, right? Now, you have neurotransmitters. When they get released at a synapse, the effect could be excitatory or inhibitory. And the neuromodulators can affect the rate at which the neurotransmitter is released. They can also affect the response at the postsynaptic neuron. And neurons can be either facilitated or enhanced, or they can be inhibited by other chemicals besides neurotransmitters and neuromodulators. And that response of the postsynaptic neuron, that can be affected by neuromodulators as well as other chemicals that can cause either an inhibition or facilitation. If there's already activity going on at other synapses, And if you change the rate that the neurotransmitter is being released through some facilitation or inhibition, okay? So that's chapter 12, that's nervous tissue. So we're gonna be moving on in the next video, or next class, talking about nerves, and particularly like cranial nerves, spinal nerves. We'll be moving into that. Okay, so but we needed this as our foundation. Okay. Also, in this unit, we'll be talking about parts of the brain, parts of the central nervous system, and what's responsible for what. Okay, or what areas are responsible for which functions. All right. And that will conclude this video.